Hi everyone, I'm Rentian. Today we'll talk about an interesting story between fungi and plants. The story is based on a paper called ectomycorrhizal fungus P. microcarpus encodes a microRNA involving cross-kingdom gene silencing during symbiosis. And now, let the story begin. Since the dawn of evolution, plants and pathogens have been embroiled in an arms race. While plants constantly develop new defense mechanisms, pathogens also equip themselves with fancy weapons to break the defense. Among these weapons are microRNA, which are small single-stranded non-coding RNA. It can be incorporated into the RISC protein, which stands for RNA-induced silencing complex. Once there, RISC will use microRNA as a guide to recognize mRNA that is complementary to microRNA and cleave it. Research has found that pathogens can transport microRNA into plants and break down the defense mechanisms. So here is our main character, the fungus P. microcarpus, and a tree called E. grandis. P. microcarpus is a beneficial fungus that can supply E. grandis roots with water and nutrients. But interestingly, research has found that this fungus, similar to pathogens, also transports microRNA into plants. But here is a question. We know that microRNA are used to break through the plant's defense. If you want to help your friend, why would you have a weapon? And this is what the paper will find out. So first of all, you need to know what microRNAs are produced by the fungus. The author used a technology called small RNA sequencing and found 11 microRNA produced by the fungus. But of course, not all of them are relevant to the interaction with plants. To find out what microRNAs are transported into the plants, the scientists separate the fungi from the roots with the cellophane membrane, which is a membrane with tiny holes. In this way, the fungi cannot physically touch the root, but the microRNA produced by the fungi can penetrate the membrane. Whether the microRNA will enter the plant depends on whether the roots will uptake them. So, what microRNAs are taken up by the plant? The scientists extracted RNAs from the roots, did another sequencing for microRNA, and matched them with microRNAs produced by the fungi. This time, they found that the plant uptook 4 types of microRNA. Interestingly, one particular microRNA called mRNA is transported in a large quantity, so there must be something special about it. The way to investigate this is to manipulate the level of mRNA. On the one hand, the scientists artificially produced both the single and double strands of these microRNA mimics and supply them to the plants. Both the single and double-stranded microRNA target the same sequence. On the other hand, they also use RNA inhibitors called anti-microRNA that can have a complementary binding with the microRNA. This will cause them to be digested. After treating the roots with either this microRNA mimic or inhibitor, they look at how this would affect the fungal development. And the result is interesting. The black bar indicates successful colonization of the roots, while the gray bar indicates failed colonization. The left 3 groups are control, while the right 2 experiment groups are supplied with microRNA. As you can see, compared with the control, supplying more of this microRNA doesn't have a great effect. They then did another experiment. This time, the leftmost group is control, and the right 2 experiment groups are supplied with microRNA inhibitors. Compared with control, you can see that inhibiting this microRNA significantly reduces the ability of the fungi to colonize the root. So this means that mRNA is important for fungus to establish its relationship with the plant. But here you might ask, what genes does this microRNA target? To answer this question, they use a program in NCBI called MEGA-BLAST algorithm to find the complementary sequence of the microRNA in a tree's genome. In this way, they can identify what tree genes the microRNA would target theoretically. They identify 3 complementary genes in a tree. But are they really being silenced in nature? The way to solve this is simple. Use a technology called qPCR to see whether their expression level drops after the plant uptook microRNA. What they found is that while all these 3 genes have decreased expression, the strongest decrease is observed in this EO3170 gene. I want to emphasize that while it looks like this LO1AA2 group also has a strong decrease, this decrease is not significant. So there must be something special about this gene called EO3170. Through bioinformatics, the author found that this gene encodes an immune receptor of the plant that can detect fungi, probably including our P. microcarpus. So back to our first question, why does the fungus target the plant with microRNA? We now know that this microRNA will suppress the plant's immune detection, but it raises another question. If the fungus is trying to help the plant, why does it need to evade the plant immunity? As you may have guessed, Not only is the fungus carrying the weapon, the plant is carrying the weapon too. Without this microRNA, the fungus will be killed before it can benefit the plant. But why does the plant want to harm the fungus if it's beneficial? That's an interesting part of nature. In nature, the difference between friends and enemies is often not that clear, especially in the case of fungi and plants. You can imagine this as a business, with the fungi providing the plant with nutrients and water, and the plant providing carbon and sugar to the fungi. Both sides are selfish and want to maximize their own interest. Along the evolution, we can find examples that the fungi may want to cheat the plant by taking more resources without giving any benefits, and similarly, the plants may want to cheat the fungi as well. With both sides having the weapon to penalize the cheater, they managed to maintain this delicate mutual relationship for thousands of years. And this is the story between fungi and plants. Thank you so much for watching my video.